2013 asking for if I might be accepting graduate students that next fall. And I was really excited because she, as you'll see, has a real genuine background in parasitology. And although my collaborators and I had been working in this host parasite system for about a decade at a time, we had this enormous black box on the immune function and the immunological responses in that system. And figuring that out had already been on the to-do list, but I knew that I really didn't have the expertise to do it. And what you're going to see is what Tara has discovered over the last 4.5 years has really transformed the way in which my collaborators and I think about that system. And it's so exciting. And as many of you also know, Tara is just extraordinarily creative. And as some of you know, she started undergraduate at UCSB as a dance major and received her bachelor's in fine arts in dance in 2008, being a professional. But then went back to school to get a second bachelor's in biology, which she received in 2011 and spent 2 years working as a lab technician in parasite ecology. And so this background of dance and parasitology and disease ecology and just this infectious scientific curiosity really brings a unique skill set and approaches, which I'm sure you'll be evident in her talk today. So she's been funded by both HIDERS and an NSF GRSP, as well as being in the last cohort of the NSF Dissertation Improvement Grants. And as of today, she has 8 published papers, including the first chapter of her dissertation, which just came out this week as the cover story in Ecology. Not that anybody ever sees the cover article anymore. Yes, but they sent a PDF of it on the cover. She has 2 manuscripts to review and another 2 to submit for her dissertation. And since she still has her GRFP in the spring, she plans to spend the next several months writing. And we were talking last week, there's somewhere between 4 and 6 manuscripts from things that she did while she was still in California to some of her side projects, to data I have been sitting on for years with So she's talking here today and defending next week, and we're all very excited to hear what she has to say, so please join me in welcoming Tara.
Thank you so much for that introduction, Carla. And thank you, everybody, for coming today. My name is Stewart Merrill, and today I'm going to be talking about my dissertation work, which is broadly focused on this one question of whether and how host immune defenses matter for the spread of infectious disease. Before I begin, I'd like to acknowledge this huge group of people that have made this work possible. First, my advisor, Carla. She's amazing. Second, all of my doctoral committee members, Zoe Rapti, Brian Allen, Becky Fuller, and James O'Dwyer, as well as my collaborators and co-authors. Thank you to all of the people that helped me in the field and in the lab. And finally, thanks to my funding sources and to the University of Illinois and Indiana University for accommodating me during this research. I'm going to start with a story today. A story from my early childhood and one of my first experiences with infectious disease. When I was 5, I started kindergarten and contracted the chicken pox. I then passed that infection on to all 3 of my siblings, which left a pretty miserable few weeks for my parents. Now, this photograph was taken in the early 1990s, and back then it was pretty normal to see infections like chickenpox in children. But we don't really see this infection anymore. And that's because in the mid-1990s, a vaccine was introduced to control this particular virus. When it comes to human infectious disease, we've developed so many different vaccines, with the understanding that by lowering an individual's susceptibility, we can prevent that individual from getting sick and prevent the subsequent spread of infection. Now, variation in susceptibility doesn't have to come from vaccines, and we know that there are so many examples where humans exhibit a lot of natural variation in their susceptibility. One of the more classic examples many of us are familiar with is sickle cell anemia and its potential protections against infection with malaria. Given the importance of susceptibility for disease, it's been embedded into what's called the disease triangle, which is a simple paradigm that describes the conditions under which disease spreads. When it comes to human, wildlife, and agricultural diseases, this paradigm states that disease results from interactions between susceptible hosts and pathogens in the environment. Now, like I mentioned, in human systems, we readily acknowledge the importance of susceptibility for influencing disease. But as we move into non-human systems, and particularly into wildlife, we see an interesting shift take place, where emphasis is placed strongly on the importance of pathogen exposure, and the importance of host susceptibility is somewhat neglected. Now, this focus on exposure is not unwarranted, as exposure is critical for the transmission of parasites and pathogens that cause disease, and this is very clearly indicated in what's known as the dose-response curve. Essentially, as you increase the number of parasites or pathogens a host is exposed to, that host's probability of infection increases. So, as a relevant example, hanging out with one friend with the flu is just a lot less risky than hanging out in a seminar room full of flu-infected people. Now, given the nature of this curve, a common and safe assumption has been that a population facing a certain level of exposure might have a predictable level of disease. But that prediction doesn't hold up if susceptibility varies. Now, we know that we can see this sort of variation in human systems, and it turns out in the wildlife system that I study, we also see tremendous variation in susceptibility. So shown here in red is the average dose-response curve, with all of the points contributing to that average coming from different populations and points in time. And some might argue that to understand disease, we really only need to know the shape of this red curve. And the question that has really motivated my research over the last few years is whether that's a safe assumption to make. In our attempts to both predict and mitigate disease, can we safely ignore all of this variation in susceptibility? I'll address that question in 3 parts today. In the first part of my talk, I'm going to unpack susceptibility. I'm going to dig into what susceptibility really is and develop a new approach for measuring it. In the second part of my talk, I'm going to explore host susceptibility as the function of an environment, considering how flexible host immunological responses are and how they can be shaped by a host ecology. Finally, I'm going to take all of this information and I'm going to move it into a natural disease system and ask how exposure and susceptibility interact to either promote or inhibit the spread of disease. So, before we can explore the epidemiological importance of susceptibility, we need to know what susceptibility is. So what controls it and how do we measure it? The answer to these questions might lie in the transmission coefficient beta. Now, when we mathematically model infectious disease, we typically encapsulate whether or not an individual gets infected with beta. It's this really nice general parameter that boils all of a host susceptibility down into just one simple term. But this generality comes with some costs. And one of those costs are that as we look toward how an individual moves from susceptible to infected, beta doesn't really tell us anything about the mechanisms that allowed that switch to occur. It's just too general to pinpoint any rate-limiting step in the infection process. But what I think is really, really cool about beta is that hidden in this simple coefficient is a complex series of events. So beta depends on the dose acquired by a susceptible host. It depends on whether or not a parasite can establish within its host. It depends on the growth and reproduction of parasites within the host and ultimately how many infective stages are produced. And when we break down beta into these constituent steps, we gain an opportunity for understanding each of those steps and what they can tell us about susceptibility. So for instance, 3 of these steps are united by a common theme, and that theme is host immunity. A parasite's establishment, growth, and reproduction can all be controlled by the host immune response. So we can potentially learn a lot about susceptibility if we can identify and quantify the immune responses that regulate these 3 steps. But as many of us know, nothing is ever that straightforward in ecology, and what I've put up for you is a basic immunological cascade of invertebrates. And I think this cascade well illustrates that immune function is redundant, it's complicated, messy, and seemingly intractable. And ultimately, all of this complexity can really hinder our abilities to generalize regarding how immune responses drive variation in susceptibility. And what I really want, what I've been working for over the past few years, is to find a happy medium, an approach to understanding susceptibility that lies somewhere between immunological complexity and the generality of beta. And in the first part of my talk, I'll talk to you about how we're using Markov models to build that happy medium. That is, how we're using Markov models to capture the essence of immunity without necessarily measuring each and every ingredient. Before I get into the nuts and bolts, I'd like to acknowledge my co-authors, Carla Cáceres and Zoe Rapti, who've both been instrumental in the development of the ideas that I'll present. Now, we study a simple one host, one parasite system with environmental transmission. In this system, the host ingests parasites, the parasites reproduce inside of the host, and then the host releases parasites back into the environment. Now, this is a fairly simple and general parasite life cycle, and we know it applies to a lot of different parasites and pathogens that are important, like the etiological agent of cholera that infects humans, or Perkinsus that infects economically important oyster populations. We study this life cycle in plankton, specifically Daphnia dentifera, that are infected by the fungal parasite Mechnicauia bicuspidata. And as Carla mentioned, when I entered this system, we knew that if you took a bunch of susceptible Daphnia and fed them these infectious fungal spores, 10 days later, some of them show signs of infection and some don't. But the within-host life cycle of this parasite was a complete black box, and we knew virtually nothing about the processes regulating this parasite's establishment, growth, and reproduction. But the power of the Daphnia system is that the hosts are transparent, and this makes it relatively easy to open that black box. So I could actually just look inside Daphnia over the course of infection, watch the parasite develop inside of its host, and quantify all of the processes that encompass beta. So now I'll describe to you what happens after an infectious fungal spore meets a susceptible Daphnia host. First, the needle-shaped spores penetrate the Daphnia gut barrier and move into its body cavity. And here you can see a fungal spore that has moved through the gut epithelium and has host immune cells or hemocytes globbing onto it and trying to kill it. Those spores then emit fungal hyphae, and the hyphae grow out laterally, kind of like finger-like extensions, and they increase in their surface area. So here you can see some spores that have gone through the gut and hyphae that are growing over them and branching outward. Those hyphae then develop into sporocysts, and sporocysts are small sac-like structures that typically reside within the Daphnia's musculature. So here we can see healthy Daphnia muscle up below, but this muscular tract but this muscular tract below has been largely overtaken by dozens of these Mechnicauia sporocysts. Those sporocysts then rupture and release conidia, and the conidia are the typical yeast form of the parasite. They reproduce by budding, they disperse through the host, and they continue budding, and this is how we see the parasite build up really high-intensity infections. Finally, those conidia elongate and form asci, and each ascus is a sac that contains one fungal spore. You can see some asci here in the host, but this image was taken using the 100x objective. And if we zoom out, we can see that our Daphnia is completely filled with these fungal asci. All of that brown material is spores, and a heavily infected animal will regularly yield in the tens of thousands of spores. So, breaking down this infected yes or infected no response and informing it with natural history has revealed these nice, distinct within-host stages. And these stages provide a great framework for thinking about why Daphnia vary in their susceptibility. So for instance, a Daphnia can limit spore puncture by decreasing the penetrability of their gut barrier. And this prevention of initial infection is what I'll refer to today as resistance. If a daphnia can't resist infection, it can kill any of the subsequent infection stages with an activated immune response. And this killing of infection is what I'll refer to today as clearance. A few years ago, I took a bunch of daphnia and I fed them a bunch of fungal spores. And then I tracked individuals through time. So I looked at each individual 3 times. I looked at them once at time point 0 when I fed them fungal spores, again at some random time between days 2 and 8 post-exposure, and then again at day 10. And here we have time on the x-axis, the time at which I examined them. We have all of the infection stages I described to you, and you don't need to worry about keeping track of those stages, as long as you remember that moving up the figure means moving through the infection process. And here, each point represents an individual, and lines track individuals. So I feed all of my daphnia spores, they move through the various infection stages, and ultimately converge on this final infection stage, the ascus stage. And I've left out a little bit of data for you, and if we include the full complement of data, we can see that some individuals moved from exposed to uninfected, they resisted their infections, and others moved from various infective stages back to the uninfected class. They cleared their infections. So now we have empirical evidence of recovery, and we know that daphnia can use these 2 defenses to kill their parasites. But how do we quantify these 2 defenses? Now with the daphnia system, with this sort of data, I could do some simple division to just calculate the probabilities with which these 2 transitions occur. But that approach isn't really suitable for non-transparent systems. If I wanted to do this in birds or frogs or oysters, I would have to dissect the host, and dissecting it means killing it, which would kind of limit the opportunity for observing infections through time. So cohort studies might be a solution to this problem. And here I'm going to show you a different population of daphnia, but rather than tracing infections of individuals through time, I'm going to track entire cohorts through time. So here we still have time on the x-axis, and on the y-axis we have the proportion of a population that's occupying a given infection state. So essentially what I do is I take a group of daphnia, I stage all of their infections, and then I throw those daphnia away, and I start the next day with a new set of daphnia. Here my daphnia can occupy any of the infection states I discussed, and I've also included an exposed state where daphnia have consumed fungal spores, an attacked state where spores are actually attacking their gut barriers. We also have a dead state to account for mortality, and an uninfected state. So at time point 0, I feed all of the daphnia fungal spores, and 100% of my first cohort are in the exposed class. 1 day later, I look at a new cohort, and we can see that some of them are being attacked by spores, some have moved on to spore infections, others have hyphae infections, and some of them have died. And we can continue tracking the progression of these cohorts through infection stages through time, and importantly, we still capture the signal of resistance and clearance as we look toward how the proportion of uninfected animals increases through time. So now we have an approach that's suitable for systems beyond our transparent daphnia. But looking at this, I still have a 9-state system, and I'm really only trying to quantify 2 host defenses. And if I want to be able to build a model that's generalizable across systems, I probably want to boil down some of this complexity. So I've gone ahead and distilled the system into 4 key and translatable states. We have exposed in yellow, infected in red, uninfected in blue, and dead in gray. And working closely with Zoe Rapti, we've developed a Markov model approach for analyzing these data. So a Markov model is a stochastic model used to evaluate transition probabilities among states within a fixed state space. And by applying our model to our longitudinal data, our model can estimate the probabilities that we're interested in, like the probability of infection, the probability of mortality, the probability of remaining within a state, and importantly for my research, the probability of moving into the uninfected class. So animals can move from exposed to uninfected via resistance, and we think that's driven by the structural integrity of their gut barriers. And animals can move from infected to uninfected via clearance, which we think is accomplished with an activated cellular immune response. Daphnia are clonal organisms, and we maintain different clones or genotypes in the lab. And here, these 6 genotypes all vary in their susceptibility to the parasite. So if I expose them to the exact same spore dose, some have low resulting prevalence and others have high resulting prevalence. And we can now use our Markov model to ask why that is, and ask whether variation in resistance and clearance are driving these differences in susceptibility. So we can apply it to our low susceptibility clone and see if it has higher resistance and clearance than a high susceptibility clone. So I've done that for our 6 clones, and here on the x-axis is the model estimated probability of resistance, and on the y-axis is that clone's empirical susceptibility, its prevalence to infection. And there's not a great relationship between resistance and susceptibility, meaning that resistance is not doing the best job of explaining variation in susceptibility. And this could be because in this experiment, I fed these Daphnia really, really high spore doses, so high that they just might not have been able to resist every single spore that entered their guts. But if we turn our attention to clearance, we can see that there's a really nice relationship between each clone's probability of clearance and its resulting infection prevalence. So for instance, our high susceptibility clone, if we look at its longitudinal data, we can see that few individuals ever enter the uninfected class, and the Markov model tells us that it has a close to 0 probability of clearing infection. Whereas our low susceptibility clone has a signature of clearance that we can now actually quantify, and it's around 60%. And we can go beyond just calculating these probabilities and apply a Gillespie algorithm to them to model what the system looks like through time. So I've done this for one of our clones, and you can see that clones rapidly leave the exposed class and enter the infected class, and then infected animals are removed from that class as they either die or clear their infections. And this is really cool to me because while I can witness within-host dynamics in Daphnia, I can't in other organisms, and by using this approach, we kind of get a window into what other organisms within-host dynamics look like. And importantly, we also capture signals in these within-host dynamics of both resistance and clearance. So this initial peak in prevalence, that peak is constrained by an organism's ability to resist infection. And as you look at the growth of the uninfected class from peak prevalence to the end of the infection process, the slope of that line is actually predicted by a clone's ability to clear infections. So obviously I care a lot about Daphnia recovering and resisting and clearing infections, but I understand that most people probably don't care about Daphnia because they're not a pressing conservation or human health concern. But this approach is meant to be general. It's supposed to be a model that we can apply to other systems beyond Daphnia, things like medically important invertebrates, like the snail Biomphalaria glabrata that transmits schistosomes to humans. My hope is that we can begin applying this model to other systems to see how and why they vary in their susceptibility and what the implications of that are for human disease. So, in addressing this question of why do hosts vary in their susceptibility, I've decomposed beta into the steps of infection and leveraged those steps to broadly quantify 2 key host defenses. And I learned a few important things along the way. First, I learned that Daphnia can recover from infection. Now, over the past few decades, there's been this common assumption that Daphnia can't recover, and these data completely overturn that result. But I also learned that Daphnia exhibit broad variation in their abilities to both resist and clear infections. Now, my data suggests that there might be a strong genetic basis to this variation, but I'm really interested in understanding whether that variation extends beyond genotype alone. Like, how flexible are these 2 defenses, and can they be shaped by the environment? In the next section of my talk, I'm going to move from theoretical work to more empirical work. And rather than focus on the Markov defenses, I'm going to use a trait-based approach to see how host immunity and host-parasite interactions respond to changes in environmental conditions. So, we all have to acknowledge at some point that with the exception of some cockroaches in Morrill Hall, labs are not natural habitat. Rather, our study species are embedded in complex ecosystems, and their interactions with the biotic and abiotic environment have the potential to influence host-parasite interactions in sometimes surprising ways. So let's consider resources, for example. We know that resources are critical for an organism's physiological functioning. And a common prediction in immunology is that a host that has access to more resources will be able to mount a more robust immunological response to its parasites. But the outcome of host-parasite interactions along a resource gradient isn't that straightforward. And that's because parasites, by definition, feed off of their host resources, so that a host with more resources might also better fuel parasite growth. I mention resources because they are not arbitrary in this system. Daphnia spend the majority of their time filter feeding, and the phytoplankton that they consume can undergo large, regular changes throughout the year. So these shaded gray curves represent phytoplankton biomass over the annual cycle in two different types of lake ecosystem. And we know that in addition to changes in resource availability, lakes can also exhibit dramatic changes in resource quality, both among and within lakes. Now, if resources influence immunity and/or parasite growth, then these large changes might be powerful drivers of host-parasite dynamics. So I implemented a simple experiment to see how Daphnia and Mechnacavia respond to low, medium, and high levels of resource availability. So here's where we get to the trait-based approach. And to define the two immunological traits of interest, I'm going to focus your attention here on spores infecting the gut, because that's where the action really happens. I talked about resistance earlier, and the way that I empirically quantify resistance is by counting all of the spores that are attacking the gut barrier, and then counting the subset of spores that cross through the gut barrier and enter the host body cavity. When considered together, these two values give us an idea of the gut's penetrability, which is a key resistance trait. Daphnia with impenetrable guts are far less likely to succumb to infection. Now, if a spore does enter the body cavity, it's met with an activated cellular immune response called the hemocyte response. And if we count all of the hemocytes that respond to all of the infecting spores, we arrive at a clearance trait, where individuals that mount greater cellular responses are also less likely to succumb to infection. So what do these two traits look like under varying resource conditions? Well, to get at gut penetrability, I've regressed all of the infecting spores against the attacking spores. Now here, a 1-to-1 line represents a completely penetrable gut with no gut resistance. But as this slope tends to zero, the resistance of the gut barrier increases. And among our three different resource environments, we really don't see any difference in gut penetrability. Now, these data to me suggest that the gut barrier and its structural integrity is a genetically predetermined trait, and that the gut barrier may just be somewhat resilient to changes in the resource environment. Now, to measure the immune response, we need to keep track and control for all of the spores that are infecting the host body cavity. So here we have infecting spores on the x-axis, and we have all of the recruited hemocytes on the y-axis. And resources do explain variation in the hemocyte response. And we see that hemocyte responses are significantly reduced in a low resource environment. So here's what I think is happening here. I think that the variation in hemocyte responses that I observed, that they vary, but that, just step that back a little bit. I think that we've hit a threshold value where once Daphnia are moved into the medium resource environment, they're really operating already at their maximum levels of the hemocyte response, and that the variation might actually get a little bit more interesting if we move into these low resource environments. So there are two host traits. Let's see how they matter for parasites. At first glance, they don't. There are really no differences in infection prevalence among the three resource environments. Now, infection prevalence isn't the end of the story. Whether a host is infected or not definitely matters for the Daphnia, because that means life or death for the Daphnia. But for the parasite, a parasite's fitness isn't just infection status alone, but also how many spores are produced from an infection. And if we look at spore production, we can see that in high resource environments, parasite spore production is significantly reduced. And what I think is happening is that all of this variation I observed in the hemocyte response, it's not really great enough to drive variation in infection outcomes, but it is great enough to drive variation in parasite growth rates. And I've done a simple back-of-the-envelope calculation to see what the implications of this are for parasite transmission. So here I've just multiplied the average spore yields in one experimental treatment by the total number of parasite infections that were produced in that treatment. And here, spore production, cumulative spore production decreases as a function of increasing resources, suggesting that resources, through influencing immunity, might be powerful modulators of parasite production and hence future risk to susceptible hosts. So in summary, resources do not affect a resistance trait, but they do affect a clearance trait. And there are consequences of this for parasite fitness. Now, all of these results that I just showed you come from a superficial laboratory experiment. And I think an important thing to consider is whether the range of resources that I manipulated is similar to the sort of variation that Daphnia experienced in the wild. And we can kind of get a handle on this by looking at Daphnia clutch sizes, which are a proxy for resources. So in my experimental data, clutch sizes increase as a function of resources. They're about 2 in our low resource environment and move up to about 5 in our high resource environment. But if we look at Daphnia from the wild, Daphnia that I collected from lakes, we see that natural systems on average promote much lower numbers of eggs and also a greater range of variation. And for me, this is a really strong motivation to go out into the field and see what Daphnia immune responses look like in natural systems. So last year, I went to Indiana, and I sampled 6 lakes over a 6-month time period. And every 2 weeks, I went to the field, brought Daphnia back to the lab, fed them a bunch of fungal spores, and then measured these 2 key host defenses. Here we have time on the x-axis, and we have, it's cut off, but average gut resistance on the y-axis in this panel, and the average hemocyte response in the next panel. And I think these data really reveal that Daphnia just exhibit a lot of variation in these 2 key defenses in natural systems. Unfortunately, I don't have time to unpack all of this variation today, but I can tell you that we have a number of projects in the works that are trying to explore what environmental factors are driving all of this variation. We're looking at things like resources, like I just showed you. We're also looking at parasite and pathogen co-infection in the field, because there are about 15 other parasites that I've found infecting these Daphnia that might be influencing their immune function. Working closely with an undergraduate, Cecily Negri, we're looking at how temperature is influencing these patterns. And I'm also working with my lab mates, Christopher Holmes and Alona Manel, to see how population genetic structure might be driving these differences. So, I've taken these 2 host defenses that I know matter for susceptibility and evaluated them under both experimental and natural conditions, and ultimately I found that susceptibility is dynamic, and how susceptibility changes in environments both through space and time might influence whether or not disease can spread in a system. In natural lake ecosystems, Daphnia experience annual epidemics with Mechnicowia, and these epidemics provide nice, tractable units of replication for disease. So here we have time, and on the y-axis we have Mechnicowia prevalence within a population, and each different colored line represents a different lake that I sampled in 2017. And my study lakes, they all differ in both the size and duration of epidemics, but they also share a common pattern. And that pattern is that epidemics tend to peak in early fall, right around the month of October. And things start to get interesting when we look at what's going on with the host immune defenses at that same time period. In particular, looking at the average hemocyte response among populations, we can see that during the peak epidemic period, hemocyte production is also at its lowest. And for me, this is a really strong, motivating field pattern to go out and ask how variation in susceptibility and immune function at the level of the individual might scale up to affect disease spread within a population. So essentially, I'm going to now bring together epidemiological data, and I'm going to bring together immunological data, and I'm going to focus on the start of epidemics. That is, when epidemics invade and emerge within a system. And using these two data sets, I'm going to identify when each of my epidemics occurred, and then ask what conditions led to their emergence. So we've made it to the third and final portion of my talk today, in which I'm essentially testing the disease triangle and asking, can we actually predict epidemics given just two pieces of information, parasite exposure and host susceptibility in an environment? So like I mentioned, last year I went to central Indiana and I sampled 6 lakes from June to December 2017, and today actually marks exactly 1 year since my sampling season ended. And I used both observation and experiment to bring together exposure and susceptibility data sets. Now, to get at exposure, I measured the prevalence of early infections in the field, as well as the number of fungal spores that I found infecting wild-caught hosts. And to get at susceptibility, I measured the prevalence of late transmitting infections, and I used infection assays in the lab to generate dose-response curves. And ultimately, I processed just under 7,000 Daphnia from the pre to peak to post-epidemic period. All right, so what can early and late infections tell us about exposure and susceptibility? Earlier on in my talk, I told you about the life cycle of Mechnicowia, and I mentioned that on average, it takes about 10 days for an individual to have a fully developed infection that's detectable. So this stage right here, the ASCUS stage, is really easy to diagnose. And this is the stage that past research has used to measure prevalence and to characterize epidemics. But now we're all in on the secret, that there are 4 earlier stages of parasite development. And if we layer those stages into our measures of prevalence, we get a really different picture of this host-parasite interaction. And we can see that a sizable portion of the population harbors these cryptic and invisible infections. And so now we can use these early versus late infections to begin disentangling exposure from susceptibility. So for instance, here I've plotted my 6 study lakes with the prevalence of infection through time, and the red shaded area represents those late infection stages. What happens if we add in the early infections? Well, we can see that in our first lake, early infections are present, but they never produce late infections and they never result in an epidemic. Our second lake has a very high prevalence of early infections that seem to correspond well with the prevalence of late infections. And we keep looking at this lake by lake, and the first thing that's obvious is that when we only look at late infections, we're dramatically underestimating the presence of the parasite in this system. And this is a finding that I've also seen in tropical plankton. But an even more interesting takeaway is that in 5 of our 6 study lakes, early infections are appearing long before epidemics actually get going. and we can witness instances where epidemics failed to emerge, sometimes multiple times. These data are really strong qualitative evidence that parasite exposure alone cannot predict epidemics. And we can further unpack this by looking at the relationship between spore exposure, or the number of spores infecting wild-caught animals, and the prevalence of infections during the pre-epidemic period. So here, early infections are tightly correlated with spore exposure, suggesting that if spores are present in the environment, they enter their Daphnia hosts and just begin growing inside of them. But we see no such relationship between those same levels of spore exposure and the prevalence of late infections, suggesting that early infections are not developing into late infections. So where are they going? I showed you evidence earlier that Daphnia can recover from infection, and my hypothesis is that before epidemics, Daphnia are just very, very good at recovering. And so by measuring their natural rates of recovery, we can explicitly evaluate how exposure and susceptibility are contributing to these patterns. And I'm going to use this lake here, Midland Lake, as an example to build my framework. So I go to these lakes and I measure the average level of spore exposure. And here in Midland, spore exposure is somewhat gradual, somewhat constant, but gradually increases as we move into fall. And I use infection assays and dose response curves to measure the population's ability to recover from infection. So recovery is essentially how many infectious spores a Daphnia can withstand while still retaining a 50% probability of recovery. It's the half saturation constant from their dose response curves. And in Midland Lake, recovery starts high and declines as we move through the season. Daphnia are just becoming more and more susceptible as we move from summer into fall. Things get interesting when we overlay the epidemic data, because the epidemic is constrained to this region of time where the average level of exposure is greater than the average level of recovery. So here I'm going to build my analytical approach for these data for you. So in this approach, we allow spore exposure in an environment to vary from low to high, and then we add in recovery, which can also vary from high to low. Now, importantly, these two values are in the same currency. They're in units of spores per individual. And the straightforward prediction is that if the average level of recovery within a population is greater than the average level of exposure, an epidemic can't start. But if the average level of exposure exceeds the individual's capacity for recovery, then an epidemic can succeed. So I just take the difference between these two values, and I call that difference delta. And then for each of my lakes and time points, I ask, did the epidemic start? And code emergence as yes or no. Then bringing together these two data sets, I ask, does delta predict whether or not an epidemic emerges? Here on the x-axis are those delta values. The purple region represents where recovery is greater, and the red region represents where exposure is greater. And the probabilities predicted from my generalized linear model demonstrate a clear pattern where epidemics become more likely as exposure matches or exceeds recovery. And we can go a step further and compete this against models looking at just exposure or just recovery. And it's really their interaction, delta, that does the best job of explaining epidemic emergence. So essentially, neither parasite exposure nor host susceptibility alone can predict epidemics. Rather, they depend critically on the interaction between these two processes. Going back to one of the first questions I presented today, I asked, is it safe to ignore variation in susceptibility? And my answer to that question is no. I don't think it's safe to ignore susceptibility when we try and understand disease in wildlife. I showed you that susceptibility and immune defenses matter for parasites in a lab microcosm and remain just as important when we move into natural field systems. Now, unlike humans, we might not be able to administer vaccines to wildlife to decrease their susceptibility, but we might be able to begin identifying natural vaccines. That is, identify environmental conditions under which wildlife operate well, where they have high functioning levels of immunity, and then take that knowledge and use it to both predict and potentially mitigate the effects of disease. There are currently 9 neglected tropical diseases that are targeted by the World Health Organization because they contribute to over 1 billion human infections per year. And 6 of those diseases are transmitted to humans by invertebrate hosts, invertebrates like Daphnia. And I recently surveyed the literature to find out what's known about susceptibility and immunity in these systems. I found some review papers, and a huge portion of the studies I found were studies documenting the basics, the functional mechanisms of immunity in the lab. I also found a number of studies that looked at invertebrate susceptibility and immunity as a function of different experimental treatments. But of the 450 studies I found, just over 10 looked at susceptibility in the wild and how that mattered for parasite transmission to humans. So the type of study that I just showed you today, that I've been doing in Daphnia, is not being done yet in invertebrates that are relevant for human suffering. And my hope is that we can begin taking all of this information out of the lab and into the field and start addressing how variation in susceptibility has consequences for the transmission of infectious disease.